It's a great pleasure to be here today, so thank you to Marty, Dan, and Dana for this network of researchers that they have managed to get funding for, and for inviting me to speak, and for Courtney for making everything happen. I'm very happy to be here. So, as I think Ruslan made perfectly clear, biomedical researchers are extremely aware of potential costs of immunopathology when undertaking their work. I mean, people who are working to understand the function of really dangerous molecules like reactive oxygen species, I think, could not fail to take into account collateral damage. However, I think that on the evolutionary ecology side, people have been a little bit slower to appreciate what immunopathology might mean or how it might operate exactly as a cost. A lot of people mention it, but not so many people actually try to measure it, or if the theoreticians conceptually explore when it might affect evolutionary trajectories for defense. So I am going to follow on from Ruslan's wonderful introduction to how important immunopathology might be, at least in mammalian systems. This is just the double-edged sword that people talk about it, right? Where there's protective but also pathological effects of mounting an immune response. I'm going to try to push further into the evolutionary ecology of immunopathology. But before I begin, I want to admit my bias is similar to Ruslan's. I think mostly about mammals and of various sorts. But for a lot of what I'm going to talk about, some great work by folks in this room has shown that insects, at least, exhibit self-harm or self-reactivity. And although I'll try to mention the relevant insect studies when I get to those bits of my talk, I may not, but they're in there. There is some evidence that insects might be doing some of this stuff too. And beyond that, I'm not too sure. To my knowledge, there is not quite so much data on other species. I'll bet the chickens, if I knew more about chickens and their TNF homologs, tumor necrosis factor alpha, one of the other big dangerous molecules the immune system uses, I'll bet we could discover that this extends to birds and beyond. Okay, so before I really get into the meat of my talk, I want to thank all the people who think about immunopathology and the evolution thereof with me. And it's quite a blend of empiricists and theoreticians. I think to some extent we're so early in the process of trying to understand how immunopathology might shape the evolution of defense strategies that we need to lean on theoreticians quite a lot. At least I do. It helps me to think about the issues. And so the empiricists most important to the work I'm talking about today are Judy Allen and Andrew Reed and Grania Long, who did her PhD with me here in Edinburgh. The theoreticians most critical for today's talk, Troy Day and Jess Metcalf, but lots of conversations with many Edinburgh-based people and beyond have also shaped my thinking, and certainly my whole gang of folks and our various funding sources over the years have been indispensable. Okay, so this is the rough outline of what I'm going to do today. I'm going to start by addressing what is immunopathology, and you already have a good handle on it, I'm sure, from Ruslan's talk, but I'm going to approach it in a slightly, I hope, compatible way, complementary way. Then move on to talk about, briefly, explicitly, about how it fits alongside other costs of immunity, except I think we don't really know that yet, so that'll be a brief part of my talk, but I'll try to tackle it head on. And then I'll move on to the theory and empirical work that allows us to assess when and why it might matter evolutionarily. Okay, so immunopathology, what is it? You can deconstruct the word. Pathology mediated by the immune system or tissue damage due to immunological activity per se, as opposed to any direct damage parasites might do. So parasites, we know, damage host tissue, use up host resources, but we're talking here about the collateral damage that these dangerous weapons of the immune system can do to uninfected cells. So, for example, cytokine storm is a phrase often used to describe an inflammatory event sort of escalates out of control, multiple inflammatory mediators, each of which quite potentially harmful to host tissue, and this is thought to be the sort of thing that's unleashed in a particularly nasty flu. Like people think maybe the 1918 influenza unleashed cytokine storms that really hammered people's lungs. Just another example, because of course the immune system uses a huge array of effectors depending on the location and the type of parasite, the anatomical location. But another example is collagen storm. I'm being sort of facetious by calling it a storm because the rate at which collagen deposition happens is much slower than the rate at which inflammatory mediators might be secreted. But the point is that excessive production of collagen around schistosome eggs, for example, in the liver, is a major driver of the pathology of that disease. So just want to also begin with this observation that I'm not saying that infections don't trigger a lot of immunopathology, but they're not necessarily the primary cause of subsequent tissue damage, and I think that'll become clearer as I talk through some examples. I also wanted to get this word up here, autoimmunity. It's kind of perplexing where the line, for me anyway, where the line lies between immunopathology and autoimmunity, except I suppose that autoimmune disease would be the analog directly of immunopathology. But I was quite reassured to see that Rolf Zinkernagel suggests that it's just when the etiology is unknown, we call it autoimmunity. When we know what triggered it, we call it immunopathology. When we don't, we call it autoimmunity. For the purposes of this talk, I'm going to leave it at that with autoimmunity, but perhaps we can come back to that later. Okay, so these are indeed very dangerous molecules that the immune system unleashes on pathogens, reactive oxygen species being among the most famous ones, or at least that have gotten a lot of attention in evolutionary ecology literature. For those of you who remain unconvinced, though, I just want to remind you about a drug trial that went horribly wrong a few years ago in London to try to convince you of the destructive power of the immune system. These were people who were receiving a treatment that everyone thought, based on animal model research, would tone down inflammatory responses, but because of some peculiarities perhaps about human lymphocytes, when it was trialed in these volunteers, within a very short period of time of being injected with this drug, cytokine storms galore were induced in these people, and almost all of them wound up in intensive care. They all did survive, but these are, quite substantial periods of time. This is now in the absence of an infectious trigger. This is just turning on the immune system, turning on the inflammatory response without regulation kicking in properly, and people were in very severe distress, and this guy lost some digits. In the presence of infection, though, this is what I think about more often. I was interested several years ago in trying to quantify, or at least characterize, how widespread immunopathology following an infection was. And so I thought I'd take the WHO's top 10 tropical infectious diseases that they worry most about, and ask whether in the human hosts, and also any rodent or other animal models for the infection, to ask what kind of evidence there was that parasite damage versus immune-mediated damage was the predominant factor. And I found that for every single one of these, the most severe cases, the people and mice most likely to die from these infections were exhibiting immunopathology. Another way to put that is that the parasite burdens were not highest in those most likely to die. It was often that the immune responsiveness was strongest, or perhaps you could say dysregulated in all those most likely to die. Just to look at those same infections in a slightly different way, this is the same list. And this reflects the years when I was doing this work, that's why it's a little bit out of date. But just to give you a sense of what the case fatality rate was for these various infections, with the exception of sleeping sickness, which really has an extraordinarily high case fatality rate. You can look at these numbers and say, well, the immune system with aid from medical science is actually pretty amazing. Those are overall fairly low case fatality rates, but these do add up to 3 million deaths a year, and an awful lot of morbidity. but I'll focus on the mortality effects today. But it's not just tropical infectious diseases that appear to have this strong component of pathology that is potentially lethal. This is a much less exotic example of colds and flu. Some nice work in various labs, virology labs around the world, has shown that if you titrate anti-inflammatory agents into laboratory animals, you can show that they're just as good at killing the virus, but they don't have quite so many nasty side effects, such as wasting and the other body condition indices that are used as an index of inflammatory response magnitude. And basically, these authors conclude that hosts exhibit a lot more immune activation than they need to clear the respiratory viruses, and that the symptoms that block up the nasal mucosa, that's my as friendly a picture of snot as I could muster, that yellow block. But a lot of the symptoms are due to immunopathology, even in common colds. So my sense is that immune systems kill millions of people per year, but these do happen via disparate mechanisms depending on which tissue, for example, the immune system is killing cells in or damaging the tissue in, and also at diverse rates. And so for most of the talk, I'm going to talk about the really acute ones, sort of along the lines of cytokine storms, septic shock at the fastest end of the spectrum, where induction and killing can both happen very rapidly. Lupus is one of the antibody-mediated autoimmunities, where the pathology tends to come long after induction, years and years, and tends to also be when it kills people, it kills them rather slowly. I think this is probably going to be very important for any sort of generality in our understanding of immunopathology, but to start with, I'm going to focus just at the one end of the speed spectrum. So in that WHO paper, which I co-wrote with Judy Allen and Andrew Reed, we sort of concluded with more questions than answers, but the overall question is, if mammalian immunity is the pinnacle of adaptive evolution, it's sort of wonderful to think about this crazy immunopathology that's so widely observed. Why has natural selection not eliminated immunopathology? We threw out a lot of possibilities in there. One possibility is an engineering issue. It's an unavoidable consequence of rapid parasite killing. For example, if a speed-accuracy trade-off operates. It's also possible that it's a coevolutionary outcome in many cases. We know that some parasites actually benefit from immunopathology. A prominent one, tuberculosis, which affects 1 billion, 2 billion people or something, right? Perhaps we're being manipulated in immunopathology. It's also possible selection has not had time to act. So I usually think of this in terms of novel environments where people tend to have too much food and too few parasites. Russlin proposed another, which is the snake venom selection. Has not yet had time to purge hyperresponsiveness to venoms from our repertoire. It's also possible its effects are inaccessible to selection. So, for example, some immunopathology is post-reproductive. I think it was Jens just had a paper come out addressing this very thing in insects, which I remember to say that, but it's a very cool paper addressing this possibility as an explanation for immunopathology. So these questions remain very much open. I'm sure we could add